Yoga. Anyway, so I'm trying to figure out how to frame the story of evolution and why I believe in evolution and why we should believe in evolution and basically how it happens. There's this tension between pop science and machine-like drawings and then the reality of what's going on in the cell. And it kind of starts at the top, and so I frame it as where is DNA and what is it doing? But first, let's figure out what we are figuring out and what we want to know. So starting at the top, we're trying to figure out what DNA is doing, which is obviously DNA is the key of everything, right? It's kind of it's where the information is written in life. And we want to figure out what it's doing in the context of living organisms and how it really works. Yeah. Okay. So starting at the top, we get life. We have organisms. We have organs and parts. We have cells and the parts of cells. And then we have the nucleus, which is where the DNA resides. Basically, that's kind of the Uber story, right? We start at, our living cells, our organism. And one of the things that we have to be careful of is that we stop just thinking about primates or mammals or animals, and that we look at the other kingdoms of life too, archaea, fungi, and prokaryotes. And it's important to kind of look at the entire picture of life when you're thinking about evolution. And then there's this, the bugaboo of information. And information runs across your entire organism, right? So information is important from when you were a zygote, how you developed, and into, your entire life basically runs on this informational stream. And then across the top, you've got information across the entire story of life. Okay. So that's sort of the overview of what the hell we're trying to figure out when you're learning genetics and you're learning biology and cell biology. So we have two dimensions of information that's important to consider. Now then we go down to cells, right? And this is a picture of a liver, part of the liver. And inside of this, there are a number of different cell types. And each one has its own shape. Each one has its own particular, what I call a proteome. It has its own mix of proteins, and it makes it the kind of cell it is. But you can see that there are many, many different types of cell in the organ, in this one little part of this large organ called the liver. And so we have all these different cells. We got hepatocytes and hepatic stellate cells, and we have Kupffer cells, which they don't picture here, and then we have proteins like collagen that run between the cells. There's all sorts of intracellular structure in your body that's made of things like collagen, and in trees, things like cellulose that sort of design the entire organism. So in any organism, I think there's about 200 to 250 tissue types in human beings. So your cells started out as this one kind of zygote, and they differentiated into all of these different types of cells. And as the organ develops, you'll see that one cell, like these little orange cells, hepatocytes, are telling the cell next to it what to do. And the cell next to it is telling the hepatocyte what to do. So the cells live in a community in these organs, and they exchange information that basically goes down, goes into the cell, then the nucleus, and it expresses a different kind of information. And so it's this interchange back and forth between cells. So what I'm trying to establish is sort of a top-down, right? Idea.
Yep. I'm listening.
How evolution develops. So here's my liver cell. So this is a picture with that shows you the I think the text underneath a little better, and shows you all the different cell types. So that's kind of what we're looking at.
So now you're talking about what kind of cell within the liver?
Hepatocyte is a good one, and this is what is actually going on in your body. These are actual cells. So you see the blood cells on the inside, and you see the basically the cells, the endothelial cells that line the artery. And then outside of that, there are muscle cells, and then there are supporting cells, and cells all the way down. And we have to be careful to consider that there aren't just the top here animal cells. We also have plant cells. And plant cells are kind of a different deal. They have cell walls on the outside of the membrane, so they're more structured, and they have more stuff in between the cells that allow plants to be rigid the way they are, right? They have these rigid shapes, whereas animal cells are a little more floppy. They flop around.
I mean, that obviously that makes perfect sense, because if, plant cells wouldn't survive, right? They would too mushy and not, they just wouldn't. Yeah.
Of course, they get mushy if you don't water them. Then they start flopping around.
Right. Yeah.
Yep. But considering many different kinds of cells, but now you can, and these are eukaryotic cells, and then then we have the prokaryotes, which are a lot simpler than this. So we're looking back maybe 700 million years ago, and maybe even a little more, to the beginning of eukaryotic cells. And that is pretty early. That's pretty recent compared to what's going on in life, because we got a few billion years that happened before that. So we're going to talk about the whole story of evolution. You have to shine that flashlight all the way back and try to see how these structures developed. Now the shock is looking at how complicated that cell in the upper left is. With all the different structures that it has. Okay, and here's a kind of on the left, we have a diagram of a cell, which is not to be confused with the actual goddamn cell, which is over here on the right.
Which is actually an image of a cell, which is not to be confused to the actual cell.
With the actual cell, right?
Yeah.
So again, we have electron micrograph of a cell. So anyway, we have the plasma membrane on the outside of the cell, and then on the inside, we've got the nucleus, and then around the nucleus is something called the endoplasmic reticulum, which is really, really important in eukaryotic cells, and not so important in prokaryotes and other types. And so the endoplasmic reticulum is kind of continuous with the membrane of the nucleus. So it's a series of stacked discs, basically. Let's see if can I go backwards? Yeah. If you see in the upper left, I'm not sure how clear that one is. You can see that all around the nucleus is this endoplasmic reticulum. And what that thing's job, you see these little dots around it? Okay, that's called the rough ER. And what's happening there is we have a ribosome. We have ribosomes that get stuck to the outer surface of the endoplasmic reticulum, and they are spitting proteins out into the inside of that, the blue part on the inside. So as proteins are being created, it's spewing them out into that little inner chamber. And there are also ribosomes out here in the tan stuff. There are also ribosomes out there that are creating proteins that end up in the cytoplasm of the cell. So you got the membrane, we got the cytoplasm, we have the nucleus, and we have the endoplasmic reticulum. So those are takeaways, right?
No, you say you want to go back further than what, 700 million years or so, right?
Yeah, but not right away.
Okay.
So we want to we're looking at evolution in the last 700 million years. And then, of course, when we start going back further, we end up with prokaryotic evolution and how this thing developed at all, right? So there's the wall between the prokaryotes and the eukaryotes, and that's one of the major talking points when you're talking about evolution. God, I can't stand that phrase anymore. Somebody keeps using that, and I don't like them. But anyway, there's this point where you go from prokaryotic cells to eukaryotic cells, and then to multicellular life.
Right.
And then we go all the way up the ladder, through the Cambrian, the dinosaurs, whatever. Yeah. And that's where we see evolution kind of springing off. So there's a story here, and it's a huge story. It's a gigantic story.
Yeah.
Okay.
Those little dots that make up the rough endoplasmic reticulum.
Yep.
You said those are
the
proteins?
ribosomes that are spitting out the proteins. Those are spitting out the proteins, right?
Yeah, the famous.
They're like protein guns.
Yeah, protein guns. Yeah, think of it as a pasta maker. If you've ever seen, one of those pasta makers that they were selling like crazy about 20 years ago, it just spits out spaghetti. Spaghetti just keeps flowing out of those little holes, that sort of thing. So that's what's going on. So anyway, backing out a little bit, okay, we have, we got cell membrane, we have the tan stuff, that's the cytoplasm, okay, and we have all of these bizarre layers called the endoplasmic reticulum, and then inside we have the nucleus, and of course we have nuclear pores, which Cell Cordova loves, that allow things to get in and out of that nucleus. Okay, the nucleus is kind of like an inner membrane, and that inner membrane of the nucleus is the inside. It's a 2-layer membrane, but the inside of it, the lumen, is continuous with the endoplasmic reticulum, which is kind of interesting. But I don't know much more about that. And then on the inside, we've got our DNA. So this is where DNA, and this is what it's doing. And this is where I showed you the chromosome territories and all of that stuff. But over here on the right, okay, well, let's see. On the right, you can see this condensed heterochromatin, basically. Heterochromatin is, how would you put it? It's other than euchromatin, which is the stuff that's getting translated. So the DNA strand on each chromosome, I think if you unwind them, they're like, I don't know, a couple feet long. So it's a really skinny molecule.
And that's from one cell?
Yeah.
Wow.
And we got 46 of them in there, 46 different chromosomes. But anyway, so on the outside of this nuclear membrane are these lamins, okay? And those are a kind of protein that's kind of sticky, and it forms a structure, and it helps to keep the nucleus in the shape that it's in. Also, it attracts chromosomes. So it attracts that DNA, that heterochromatin, condensed heterochromatin is stuck around the outside of the nucleus. And then pieces, you imagine a ball of spaghetti that, if if you throw your spaghetti in the pot and you let it cook, it forms those big balls if you don't stir it or put oil in it. Well, it's kind of like that, and yet it has all these strands coming off of it that are poking into the center of this nucleus. And the strands that are coming off of it and these, these looped structures are the places where the actual transcription is happening. That's where we're taking the DNA and we're creating messenger RNA, basically, on those spaghetti strands that are sticking in the middle. Now, a part of them, and I think particularly on the ends of chromosomes, seem to gather together in this center called the nucleolus. And this goes back, and this is part of the story of evolution. Those ends what they do is they create the ribosomal proteins and RNAs in the nucleolus, okay, along with a number of other things. But there's a lot of RNAs that are being created that don't end up being proteins that are created in this nucleolus, but it also creates the ribosomes there. And now it doesn't have a membrane. It's sort of a gathering place. And you can see on the right that it stains differently. Okay. You got the nucleolus. And it was just a curious structure that it took them a while to figure out what exactly was going on there. But
so there's no real membrane around the nucleolus. It just is a clump of similar material.
Yeah. Yeah, and along with a whole lot of a cluster of other proteins that are involved in creating those ribosomes and creating all of the RNAs that end up doing very complicated things in the nucleus and in the cell. So we're creating a bunch of RNAs there that never get translated to proteins, and some of those RNAs are at the center of the ribosome. And then there's other proteins that attach to the ribosome. But the whole point of this nucleus is to create mRNA, the messenger RNA. And that messenger RNA gets transported out into the cytoplasm. And those are the little dots. The little dots are the ribosomes that grab onto that mRNA and they start producing proteins. Now, what they don't show you in this diagram is that some of those little dots are floating around in the tan part. So there's two places that proteins are being created. One of them is on this rough endoplasmic reticulum, and another one is in the cytoplasm. Now, when a protein is created, it ends up being processed further and delivered to different places, okay? So we have transcription inside the nucleus. The mRNA goes out of the nucleus, and then it becomes translated into proteins on either the endoplasmic reticulum or in the cytoplasm on the outside of the cell. Okay?
Yep.
Also, and you got to be really solid with that part of the story. Okay. And then, let's see, what do I have here? Now I got another picture of a cell because you can never have too many goddamn pictures of the cell. So in fact, we have two pictures of the cell. Okay, the one on the left, a little more complicated. You see the endoplasmic reticulum.
Mm-hmm.
And there's also smooth endoplasmic reticulum, which is sort of a factory where other things are done. But now the proteins that are being created on the surface of the endoplasmic reticulum end up spewing out into the inside and being transferred into the center of that ER, those little lumens in the center of those discs. So we're looking at you got the purple part, which is the nucleus, and then you can see the pores on the outside of the nucleus. And then the blue part is the endoplasmic reticulum. And proteins are being spit out into that. And from there, they go through a big journey. Like they have certain signal sequences on the proteins themselves that, oh my God, I should have backed up. Okay, these signal sequences do a couple things. One thing they do is once the ribosome attaches to that mRNA and starts producing a protein, they tell it to stick to the outside of that rough endoplasmic reticulum. So it gets stuck to the outside because of a signal sequence. And then it goes on the inside, and further signal sequences kind of send it to different parts of the cell. And one of the really important parts, so we're looking at, okay, we transcribed some DNA into mRNA. It's gone out here. It has a signal on it that allows it to get stuck to the ER, the endoplasmic reticulum. It produces the protein. It goes on the inside. From there, it gets processed a little bit. Some chemistry might happen, and it goes out to something called the Golgi apparatus, which is the kind of, what color would you call that? Is that pinkish brown?
We'll call it pinkish brown. I'm sure there's a single name for that, but I don't know colors that well.
Yeah, I don't have I used to have all the color cranes. Like I had the 128 color set, but I never understood their names. I don't know. Anyway, so it goes out into the Golgi, and the Golgi is like a stack of pancakes, but it also has like an inner lumen, just like the endoplasmic reticulum. In fact, they're kind of closely related in the composition of the membranes, but just a little bit different. So starting on the inside of the Golgi apparatus, the first pancake is called the cis area. Cis means near, and it goes all the way out to the trans area, which is after each of these little chambers does its little bit of processing and sends the protein onto the next area. And then when it goes out to the trans area, parts of this will bud off, and it will merge with the membrane of the cell itself. And that's how proteins get out into the membrane. And they're floating around on the membrane, basically. And also, the Golgi apparatus is responsible, along with a few other parts, for producing the membrane itself and creating membrane phospholipids that form that bilaminar, outer membrane of the cell. So that's how the cell gets maintained. So there's kind of the whole story from transcribing to translating to getting out into the cell membrane. Okay. And let's see, what do we got? What else we got here? Plasma membrane. We got the mitochondria, which are pretty famous on their own. We have flagellums. Cells have parts. Then we have the smooth endoplasmic reticulum on the upper left, which I'm not going to talk about too much right now. So,
now, do all cells have a flagellum?
No.
Okay.
A lot of them, a surprising number of them do.
Okay.
They have structures that stick out of the cell, and that's where we go back to this, this liver picture, right there. There are many different kinds of cells, and each one has its own weird little structures. Like in your intestine, there's these little villi, villi, that poke out and digest your food, so those are kind of, little appendages on the cells, and every cell has its own unique shape and structure depending on, and we're going to back out in a minute here to what I call the proteome, I think. But I want to show another picture.
Is there a big difference between cells in the liver that, are responsible for filtering out toxins, and then the cells that are processing alcohols into sugar or carbs into, I don't guess they process carbs, but that kind of
there's the liver has, I think, about probably about 24 specialized kinds of cells that do different things, create fats, and that's how your liver gets fat, and they regenerate new liver cells, and there's just a lot going on there. But I didn't want to get too far into discussing one organ, but that may be a way to go. We should probably pick some organ to talk about at some point, but not yet. But now I have another picture of a cell because you can't have enough pictures of cells.
I agree.
Yep.
Now, what this looks like is around the the nucleus, is that the endoplasmic reticulum?
Yep, the blue stuff with the little
And
red
then
dots
the
on it.
green stuff, which I can't remember the name of.
Golgi apparatus.
Yeah.
Oh, actually, that was going to be my guess. They look like somebody took, those thick, wide rubber bands.
Yeah.
That like they took a real long length of rubber band and just kind of wrapped it around there,
Yep.
and then squished it.
Stack of pancakes.
So if you took like you were saying that, maybe that was the way to go, is like take one particular organ, right? With that, I mean, if you did that and stuck with just that, easier to explain most stuff, right? That
Yep.
would be the purpose of that. Now, and that would generally not 100% apply to everything else, kind of, sort of.
No. It would apply to
like as far as like the basics on how cells operate. I don't know. So I don't know.
Yeah, they all have a common core: the nucleus, the endoplasmic reticulum, the Golgi apparatus,
Right.
lysosomes, mitochondria, all that stuff. But each one is going to have its own shape. And as you look at the whole story, the important thing is to not start to picture your whole body as just being this one cell type, right?
Right.
And in fact, on this cell type, we've got some other weird structures, and I don't have a caption for this. I wish I remembered where I got it. But this has these little villi that stick out of the cell, right? The cell membrane is not just a big ball, but, it's got little fingers that stick out, and it's got structures on the inside of it that are probably similar to Golgi structures. And so there's a lot more going on here, right, with the shape of this cell. And then we have square cells, and we have triangular cells, and we have amoeba-like cells, and, there's just a whole range of tissues. So you don't want to get stuck with you don't want to let the diagram mislead you into some sort of reductive, simple way of looking at this, right?
Now, what's that nipple ball looking structure there? I guess it's yellow goldenrod on the bottom of that picture. It's on the inside of the cell. Yeah.
That I currently do not know.
Okay.
Because I think there was no caption for this cell. I'm not sure where I found it. But I'll get that, and we might edit this part and stick something in there on what that is. So
looks like I want to know those big. They have these things on the playground for children that they climb around in. They're enclosed like that, and like it's holding pasta or something. Yeah. It's weird.
Yeah, it looks like it's got some microtubules in it and some structure, and I think it might be the centriole, the part that helps the cell divide. Mm-hmm. It could be stuck out there, but because that's around here somewhere. So, and then is this purple pipes that are going into the villi.
That's the Wi-Fi.
Yeah, I think that's the Wi-Fi. Yeah, those I don't know either. So I got to find a caption for this and maybe go a little further into that. But let's see. And just in case we haven't seen enough pictures of the cell,
no, everything's orange.
Yep. Now this one shows real nicely. You see the nucleus has 2 layers around it, right?
Right.
And it's got little holes in it. And those are the nuclear pores. And then, of course, we can see the nucleolus on the inside. But you can see where it goes into the endoplasmic reticulum, that the inner part of the endoplasmic reticulum is continuous with the in between those 2 layers on the nucleus. Okay? Can you see that?
Yeah.
Yep. And that's could be important. And then we have where's the Golgi apparatus? Okay, here's the Golgi apparatus, and that looks like a nice little stack of pancakes. Mm-hmm. And it isn't always that pretty in the cell, but it kind of gives you an overall idea of the organization. You go from the cis to the trans, and then it will deliver stuff out into the cell membrane. Now, some of the things it delivers into picture a bubble. When you blow a bubble, a big bubble, right? And it's kind of shimmering in the sun. You know how you can see liquid kind of floating around the outside of the bubble, like it shimmers?
Hmm.
You know what I'm talking about? Okay.
Yep.
So that bubble, and you can see that shimmering and that moving around. Well, some of the things that get delivered out into the membrane are these transmembrane proteins, these pores that help the cell to communicate with the outside world and to have ions go in and out or signals go in and out of the cell. But those things are floating on rafts. They found out that the dynamic structure is that these things are kind of floating around the outside of that bubble, and they're floating around a lot. I mean, they're moving, bumping into each other, and they're moving along. So they look like a one of those sailboat races they have, except nobody knows where the finish line is. Everybody's going every which way, and you got sailboats floating around on the surface of the cell. And that is a fairly dynamic structure. And there are a lot of them. There are a lot of things out there. So that's one of the things that the Golgi does is it delivers those things out there. And there's basically a piece of that Golgi will get cut off and it will merge with the cell. So the pore in the cell, the channel, they call it a channel, it gets created in the Golgi, it gets pinched off and it goes out and merges with the cell. And each of these things are like a little boat floating around, waiting for somebody to signal it, right? Like for instance, if you have a cell that's involved in sensing smell, smell cell, smell cell. That sounds like a terrible cell, but a smell cell. It will have a protein that's floating around on the surface, and then when a molecule from a lilac comes and attaches to it, it will send a signal to the inside of the cell. So cell signaling and delivering things in and out of the cell, ions, salt, sodium, potassium, all that happens through those pores, and that all gets created with proteins and such. Okay.
Yep.
Now, one of the other things that the Golgi apparatus does is suppose you have a gland that has to secrete like sweat or mucus or whatever. Those things will be created in that Golgi. They'll go out and they'll merge with the cell membrane and they'll spit exocytosis, they call it. They'll spit their contents out into the fluid around the cells, into the extra or intracellular space between cells. Back to the liver cell, or the liver with all the different cell types. There are many different cell types, and some cell types, their only job is to secrete shit. And that's why you have to blow your nose occasionally. It's a real problem for us mammals.
That's true. But then again, without it, we'd be up shit creek too.
Yeah. And let's see. And you can't have too many pictures of a cell.
I agree.
Okay, this one shows the nucleus, which we're homing in on, right? And you can kind of see how, now this gives you a better idea how the DNA is, the dark purple DNA is sort of, it gives you a little bit of an idea how it's organized, but it's got a lot of strands just floating around in the nucleus. And then, of course, it's got its chromosome territories and the nucleolus in the middle. And then it shows you the endoplasmic reticulum again. Okay, so let's see. It's a little bit on the endoplasmic reticulum. An extensive network of closed, flattened, membrane-bound sacs called cisternae. And smooth endoplasmic reticulum is smooth because it lacks ribozymes. It's a site of synthesis of fatty acids and phospholipids. So it's like a little factory. It's one of Otangelo's factories.
One of them factories that proves God.
Yep. And newly made membrane proteins remain associated with the rough ER, and the proteins to be accumulated, secreted, accumulate in the lumen, the aqueous interior of the organelle. These vesicles, which bud from regions of the rough ER not coated with ribosomes carry the proteins to another membrane-bounded organelle, the Golgi. Okay. Now, here we got a fucking mess. So now the whole point of this is that every cell is different. Every cell has its own shape. It has its own particular composition of organelles, and it has its own proteome. In other words, if you look at all the proteins that can be produced from your DNA and you put them in a spreadsheet, and on the right, you write the percentage or the number of protein molecules for every cell, okay? And then every line on that is basically a protein. And for every cell type, you're going to have a new spreadsheet for how many, like it'll have 3 molecules of this. It'll have 1.5 million molecules of this and whatever. So every cell has its own proteomic composition. Okay. And it ends up being a big, what I call puddle of mud. And the reason I put this diagram here is it shows, I think, Raw Nakedness, Stupid Whore Energy helped me find the paper for this. But this is how I think the mechanism that allows the DNA to organize itself on the inside of the nucleus. So this has something to do with that. I put this diagram up to confuse, to show you how complicated the interactions of all of these different things are. And we're not going to get into any details on it. And now here's a more simplified diagram that shows you a pathway. And I believe this is part of what they call the mTOR pathway. And this just shows you a little bit of, you'll find these in biochemistry books everywhere. And these are the things that lead the creationists to believe that it's a fine-tuned machine. Okay? But that right there, that's not a fucking fine-tuned machine. That is a ball of mud. And that'll become important later when we talk about the story of evolution, I think. Okay. Now, the next thing about this, we were talking about these things that poke through the membrane. Okay. They're up on top. You see that those 2 layers of blue dots, which are phospholipids, each one. Mm-hmm. Okay. And then we've got these membrane channels that go all the way through the membrane. These are the boats floating around on the membrane of the cell. And they allow signals to get in, and they signal stuff on the inside, or vice versa. They can actually signal stuff on the outside in different ways. But it's very complicated. So, and there are, if you put all the pathways, we would end up with an entirely black screen because there are so many pathways. So complicated, right? Here's another cell. This one shows cell growth, and this is a more machine-like reductive diagram showing you an activating ligand or chemical cell surface receptor goes down through this chain. I want to point out here, when you see these diagrams, you see the arrows?
Yes.
I see arrows. Okay, you see the funny little T-shaped things?
Yes, I see T-shaped things.
Yeah.
Okay. An arrow will be something that basically P13K up here will activate PDK1. If we're looking at the upper right, but P10 will inhibit that activation, basically.
Okay.
Oh, yeah.
And so we have activators and we have inhibitors.
Yeah.
Right. So what we have here is if you look at logic gates on a computer, right? You have something that will you have a 1, which means it's going to activate it, or it can invert it and turn the 1 into a 0, which means it's going to inhibit. So on the top here, we have P13K activating PDK1, activating AKT, which shuts off these 2, TSC1 and 2, which shuts off RHEB, which turns on mTOR, which goes down and branches out, and it turns on SK61, but it shuts off for E-BP. So there's your logic gates, right? When they talk about the cell and information being a computer, this is what they're talking about.
Yeah.
And they have, unfortunately, there's a laboratory that makes these diagrams, that's involved in this. They get more complicated, but this is a nice simple one that shows you activation and inhibition. So inhibition is the upside down T. So it's a NOT gate, if you're familiar with logic, sort of thing. So when Maddox talks about how it's like a computer, this is what he's talking about. That we have one thing activating another. But when you think if it's like a computer program, why would a programmer say not together, which means do it, right?
Mm-hmm.
So don't do it is how it works out. So that's the logic gate. There's a lot of that shit going on in cells, and it sort of gives you the idea about how this evolved by a bunch of things slamming together until we got something that worked. And that isn't how you program. You program from the top down. You use your intelligence to design the program, and you never say, don't do this, but do that. That's
Right.
And then,
yeah.
And gates would be if we had a branch coming in and two arrows had to activate one thing, that would be do this. And if you have this and this, then you have this, right? Then it activates. And then, an OR gate would go either way. It would kind of split. And it's kind of hard. I mean, it is a stretch to turn this into computer logic gates, I think.
Yeah.
I think it's a good stretch.
When you're trying to learn this shit, right? And, I sit here and I, try to learn this stuff as a, as an atheist, I guess you'd call it, right? And to me, it's very simple. It's fascinating to learn the natural process, and I don't think of God anywhere in here. Like, what am I supposed to? So then you try to put your creationist cap on, right? And you try to think on their mindset, and I'm trying to think, well, where the fuck is God? I mean, he's just, I guess the way they see it, he's just, it's so complicated, it has to be, right? And because there's information being translated, it has to be a mind that translated that or programmed that or whatever, right? I mean, that's it. I don't know.
Who's that Catholic guy that was on Germania last night?
Canadian Catholic traditionalists.
Catholic traditionalists. Okay.
Yeah.
So I think it was Simos that was trying to, was trying to make a point to him and argue with him and saying, okay, when you start with the zygote and you go all the way from a zygote to a fully formed human, there is no godly intervention there.
Right.
Okay?
Right. That's what I mean. It's like, where do you trying to,
and they won't admit that.
Right.
I was on
Smokey's
channel with that Anthony, that madhead fucking Anthony who we haven't seen for a few months.
Dipshit. Yeah.
Yeah. And I was trying to tell him,
Anthony, right? Tony Maurice. You talking about? Yeah.
I was trying to tell him that I was trying to get him to admit that there is no God intervention from the zygote to a fully formed human. That's one thing to think about.
Right.
But they keep wanting to talk about, and so did that Catholic guy last night. He kept wanting to talk about how did the information get there to begin with?
Right.
What happened before the zygote? How did the zygote get created? And Simos was trying to say, at least focus on we've got a zygote. Now we go all the way forward to a human. That is a miraculous thing when you think about it.
Right.
Because it happens with all these pathways I'm showing here, right? And nowhere in this is all chemistry. And these proteins have to, we'll get to that in a minute, and then I think we're going to wrap up for this session.
Yeah. I'm about to crash.
Oh yeah. Have I filled your brain?
Well,
So,
my brain is tired. It started out tired this morning, and
okay.
it's just continually starting to. I'm
Mine
probably going to
too.
have to take a little snooze later.
So when you
Okay.
talk about, so one point I just wanted to bring up too is that, you know, you look at this and obviously extremely complicated, right? All this stuff,
Yep.
the process and all that. And it's like when the argument would be is you have this all powerful being that they claim that exists that can do this. And of course, you can come up with your imagination or use your imagination and say, you know, why the fuck did he, if he can create this or he can create universes with just his mind, he could have made it a hell of a lot more simple and a hell of a lot better than what it is. Far less suffering, you know, I mean, it's very easily to take all, you know, we're talking about all these cells, right? You put all these cells together, create a human being, and it's very easy to grab that human being around the neck and choke him to death. And that's it. You're
Yeah.
done. Like, you know
Yep.
what I mean? Like, what? It's so easy to die. And, you know, you're so vulnerable to the environment and whatever. You know, why does it have to be that shitty? He could have done anything
Yeah.
possible, right? It's just,
If just one of these little balls, what are these little connectors? If one of these little inhibitors or activators goes to hell, you get cancer and you die.
right. That's what I mean. It's like,
Just one.
why, right? Why does it have to be that way? This all powerful being can actually create this. At least you think he can. He
Yep.
could have done a lot better and a lot more simple. Like this, you can go on and on and on with that argument. Like,
Well,
It's
here's
just
the
strengthen
thing.
your faith, my son.
yeah,
Here's the
that's
here's the thing they want to say, that God created okay, suppose every time there was a zygote, suppose there was no such thing as sex, that every time a baby was created, right? That God created the zygote. He miraculously made a zygote with all that DNA in it, all that information, and planted it in the mama. And then it turned into a human being all by itself. So God created something. There was a miraculous intervention point at the zygote. And it went from the zygote to a fully formed human naturally. So that was all stochastic, natural, random, molecules bumping into molecules, random chance bullshit, right? And no Christian or creationist even will deny that there is no miraculous intervention from zygote to fully formed human.
Yeah.
Okay, you could back them up and say, well, what if the zygote is a miracle? But then no, they don't want to do that either. They want to back it all the way up to the zygote came from your mommy and daddy, came from your grandpa and grandpa, or your grandma and grandpas, and all the way back, right? All the way back to Adam. But for animals, they have a different story. They have all the tigers evolved since the ark, right? So they're looking at miraculous intervention 6,000 years back, not at the zygote. And the thing is, put a pin in it as to where you want the miracle to occur, right?
right. Yeah.
And that's going to become a really important thing that we need to understand and talk about, because this goes in many different ways. Like they want to say, like if you start talking about evolution of primates, they'll want to talk about evolution of fish to reptiles. And then if you start talking about fish to reptiles, they'll start talking about prokaryotes to eukaryotes. So where the fuck do you want to put a pin in it and start this story? So anyway, this narrative that we're going to do here, we're going to try to put a pin in each part and talk about all the different stages of it is what I would like to do. That would be the story of evolution. The thing we aren't going to do is move the fucking goalposts. Every time we put a pin in something, you can't put a pin somewhere else, right? If you put a pin in the eukaryotes, they want to put a pin in abiogenesis. If you put a pin in abiogenesis, they want to put a pin in planetary and stellar formation. And Hovind will go all the way back to the Big Bang and call that evolution. And it's like, We got to settle the fuck down and figure out what's going on. But the one thing to get them to admit out loud, and they will never say it out loud, they'll go from one thing to another, that the zygote turns into a human being, or a zygote turns into many other animals and plants and fungi, all by itself, naturally, stochastically, through entirely natural processes. They won't admit that out loud. Try asking them. They'll keep. They want to change the question. They want to say, but
Yeah.
yeah.
Yep.
Okay.
Gish
But
Gallop.
that's that is the most important part, because what happens from the zygote to the fully formed animal, plant, or fungi, or whatever, is the same process that happens when you go from rocks on a sterile, non-life planet all the way to where we are right now. The same processes, same natural, stochastic shit bumping around process. And what I wanted to show you here today is how much shit is bumping around, and that's why I showed you the complicated diagrams. Okay.
So the aim of the lesson was to show us lots of shit bouncing around, and we have learned that there's lots of shit bouncing around in the cell.
But now we want to bring it all back to the beginning, and we want to back to that diagram I showed you with the life, the organs, the cells, the organelles, the nucleus, the DNA, and what is the DNA doing? Okay, at the very top now, this proteome, okay, is they have a number of things: transcriptomes, proteomes, metabolomes. We've got ohms up the ass. We got a lot of ohms. Okay? But this is sort of like every cell has a spreadsheet in it of this particular things that are transcribed, and it's going to transcribe them either into mRNA or RNAs, right? So it goes to RNA. And at the top, we have the genome and the DNA, and that's where the thing begins, and it goes down, and then it creates a certain set of proteins, and then it creates a certain set of metabolites, sugars, nucleotides, amino acids, ions, whatever. And all of that turns each cell into what it is, okay? It goes from all the way back to, and I could have just clicked on the goddamn thing right there. So all of these different cell types, everyone has its own specific metabolome, right? Its own specific composition. And all that sort of comes, well, it comes from a couple of different things, which we'll talk about next time. And we'll talk about how the cell gets to be one of those particular liver cell types. Because there's more to it than just the DNA. And I don't know. So I think that filled your brains up.
Definitely.
My brain is filled up for the
Remember
day.
the endoplasmic
Process.
reticulum, the Golgi, the nucleolus, the organization of DNA. And at the very top, the last thing and the most important thing I want you to think about is this little compositional diagram we're looking at right now with what exactly the cell consists of. It's not a machine. It's a bag of different varying amounts of all of these things together, and they self-organize into whatever is inside.
We got bags. We got bags of molecules.
So yeah, so I mean, like you said, you want to try to put a pin in it, try to pin them down, but like you said, that that'll never happen because of the fact that they'll just come up with some sort of an excuse of, well, I can't explain God's mind, and I'd have to be God to be able to explain that, and that, so those are the kind of excuses you're gonna get. But
they don't want to listen to our story.
They don't.
Story,
right.
They want our story to have a gotcha plot hole that goes all the way back to the producers, where the producers deny that we're going to make the movie at all.
Yep. Yeah. Exactly. Yeah.
They're like, we're not going to make that movie.
It's always this. Oh, I can't explain it all. Listen to Aunt Angela. Well, I can't explain the mechanism. I can't explain the explanation. It's already explained in whatever. This bullshit.
Yep.
So, yep. But yeah, it's like, and you look at it as with a creationist mind, is like we said, well, why the hell are this all? What was the point of all the extinctions? Right? You got friggin' extinctions. 99% of all things that have come to be on this planet has gone extinct. What's the purpose of that? Like,
Yeah.
well, couldn't you just make it all and just let it, just let it go? It's no, yeah. There's so many holes in it that it's just.
That stuff we did on Georgia Purdom yesterday on the weather report is really important because she was walking a fine line and lying to herself about, okay, everything was created perfect. Everything is functional today in the genome, yet there's mutation and things have been falling apart ever since. And we have genetic entropy. So when they try to put genetic entropy together with the perfect creation, together with 100% functionality, they have a fucking problem. They got some explaining to do, right?
Yeah.
I mean,
And
if everything
there you go.
mutated.
Yeah. There you go. That's a perfect example of, like, they have to like lie to themselves, and it's hard to separate and say they have to know that they're lying to themselves, and maybe deep down they do, but they just ignore it. I don't know. It's so hard to
They
figure
see
it
us
out.
as doing the same thing, that we're going from, they like we have these picture boards here, right? And there's a lot of details in the cracks here that we haven't addressed
that we don't know.
and they're saying, oh, those things can't happen, therefore the evolutionist is lying to himself. But I want to bring you back to that idea that the zygote, okay, even if it was miraculously implanted, the zygote turns into a fully formed organism without any miraculous intervention through entirely natural, stochastic, random chance means, right? So once we have that first cell, it turns into the organism all by itself. And that is a tremendous miracle. When you look at embryology, this is like a great miracle. It's unbelievable. It's like you look at it and you say, oh my God, God must have done that. God must have turned the zygote into a fucking elephant, There's no way it could have done it through natural means. But everybody agrees that it does. All of us, modern humans, agree that there's no miraculous intervention in the zygote. So there you go. Want to wrap it?
Let's wrap it up.
I think I think this was, well, we'll go backstage and talk a bit about this.
Sure.
Okay, so I'm going to end it. Well, we're not going to.
All righty. Thank you, Speed, for taking the time to show this to us.
Thank you. Great. Fascinating.
Let's see.
Yeah. And you do know the dinosaurs were on the Ark, though, too, right? Those were definitely on, because I don't know if you watched the debate last night with Erica, but Batman and Erica had a debate last night, and Batman made that very clear. Yeah. He made that very clear. Yeah. That the dinosaurs were on there, yeah, on the Ark. So
how many dinosaurs?
I just want to make sure. He said less than 200.
That's a lot.
He said he claimed that there was between 60 and 80 kinds, as you know, they call them. And then, which means that would be, double that, male, female. That would be less than 200. So between 120 to 160 actual dinosaurs. And they were very small. They were only
babies.
Yeah, they were. Yeah. No, did he say no larger than a hippo? I think so. I think that's what he said. I can't remember now.
Right.
But he was saying very small. They weren't big. And then he also touched on the meat that now he was not saying they weren't plant eaters. He actually claimed that, it's no problem to explain that they dried the meat. They had dried meat.
Okay.
For the carnivore.
Yeah, because that was after the fall, Yeah. Noah's Ark. So they were eating. They were eating things, and then when they hopped off the Ark, what happened to them? They eat each other. 'Cause
You know,
they just
I didn't watch the whole thing actually. I
they just
watched some of it. It was just, but I did catch that part of it. Yeah,
they disappeared very quickly.
yeah.
So at the same time we had radical speciation, we had radical extinction
Yeah.
going on.
I think they got all killed off during the Civil War because there was war going on at that
Oh,
time.
okay, so they lasted
Remember?
that long.
Yeah,
That's
I think so because I mean, according to Matt Powell, I mean they were shooting them during the war, so I think that's when they went extinct. I don't know.
yeah, so Darwin must have found a few dinosaurs when he was
Yeah,
like
he must,
living dinosaurs when he was floating around
he must
on
have.
his
Yeah,
on his boat. I just wonder why he had such a problem talking about them.
yeah, I know. It is a mystery. Some of this stuff. It's it's mysterious, but God can explain it all. I mean, we know that.
Yep.
That's just something that's just an absolute given. Yeah.
It's kind of weird because when I was growing up hearing creationist stuff, they were content that the dinosaurs went extinct during the flood.
Yeah. Not Batman.
Yeah.
I was just referring. I don't know if you heard. I was just referring to Erica and Batman had a debate last night on modern day debates.
Yeah, I didn't get a chance to check that out.
Yeah, he was talking about 60 to 80 kinds of dinosaurs were on the ark.
Yeah.
And they were no bigger than a hippo, not really taking into account what a problem that is. So,
have you ever tried to lift a hippo?
they're pretty thick, aren't they?
Yeah, I
Yeah.
would say so.
Yeah.
That's pretty big. Yeah.
Yeah. Pretty hard to lift. Anyway.
And you would have to take the meal equivalent of dried meat, quote unquote, for a whole year or more with you. So that
Right. That's
because when you get off the ark and there's only two of each of that particular dinosaur,
They better
and
have full
other dinosaurs,
stomachs.
where's the prey going to come from other than other small groups of underpopulated animals?
right. I know.
Well,
It's just amazing.
that would explain a lot of the extinction, with dinosaurs. They all ate each other, and then eventually the tigers ate the dinosaurs. So that's why they disappeared so rapidly.
Yeah.
And why the Greeks don't talk about them and the Romans. And, I don't know. I just think if they really think about their story in detail, their story has holes in it. It has plot holes.
Yeah, you think?
Yeah.
Yeah, I'm thinking. Yeah, definitely.
Yeah. Roughly the size of Suicide Squad.
Yeah, you have to. Yeah, you really have to jump through a lot of hoops to make it work.
Yeah,
I would say. Got to be Michael Jordan, I guess you'd call it, jumping through a lot of hoops.